University. His neuropathology residency was at the University of Toronto, FRCPC, in 1993. He has worked as a diagnostic neuropathologist and clinician scientist since 1994. He held the Canada Research Chair Tier 1 in Developmental Neuropathology from 2004 to 2017. He chaired the Specialty Committee in Neuropathology within the Royal College of Physicians and Surgeons of Canada from 2008 to 2014. He then served as president of the Canadian Association of Neuropathologists from 2015 to 2018. He is currently a vice president for the International Society of Neuropathology and one of the senior associate editors for brain pathology. His clinical interests are in pediatric neuropathology and forensic neuropathology. His research interests include the pathophysiology of brain damage caused by hydrocephalus, hemorrhagic brain damage, and effects of substance abuse, especially alcohol and volatile solvents. And I'm really looking forward to this talk.
Okay, thank you. Presume you can hear me. So when I, so just, there we go. So there are my disclosures, some small royalties for textbook sales. So when I was assigned this topic, I say assigned, it's because it's something that most of us don't see much of anymore and don't really think about. And I wondered, okay, so what can I do beyond just summarizing what's in textbooks? And so I looked at textbooks of various sorts and realized that a lot of them have lots of detail about genetic frequency and details of different diseases, but there's not much coverage of this topic of storage diseases and metabolic diseases in a broad context. And I'm going to focus eventually on the storage diseases, but before I do that, I'm going to give a background about why these diseases even occur. And so this is a simple diagram from a recent review paper. Genetic mutations can give rise to abnormalities in enzyme activities, and in some circumstances that could lead to a cancer, for example, and in other cases that can lead to metabolic disorders. And there are numerous classes of metabolic disorders. This lists 5 of them. The urea cycle, glycogen storage, lysosomal storage, tricarboxylic acid cycle, and mitochondrial fatty acid oxidation. And these disorders can all lead to brain disease. And that's a very simplistic view, but the reality is this. So for those of us who are old enough and who took, say, biochemistry in pre-medical school, a lot of labs had the posters from the Roche biochemical pathways on the walls, and they were extraordinarily detailed. Now, the Roche has indicated that they will either be updating or perhaps discontinuing the online version of these posters, but I've given a couple of references to alternate views of this. Basically, these posters show the interconnection between most of the known metabolic pathways and cellular or molecular processes in primarily mammalian cells. The metabolic pathway poster can be broken down into different regions. So on the top right, for example, you have the purine metabolism. Top left, you've got carbohydrate metabolism of various aspects, amino acids, the TCA cycle for producing ATP, and amino acids down on the bottom right. And if, for example, you zoom in on the tricarboxylic acid cycle, these are the details. So it'll have, these posters have the chemical structure and all of the enzymes involved in these various pathways. And so it's horrendously complex. And as far as enzymes go, the various databases list well over 3,000 enzymes in humans, most of which are non-digestive, and those are a few websites where you can look into that. Now, as far as diseases go, the most comprehensive listing of them, not just listing, discussion of them is in the Online Mendelian Inheritance in Man, the OMIM database, which if you ever use, you'll realize every time you log in, they ask for money, and it's because it's difficult to keep this updated, and they're not always having a steady funding stream. There's over 25,000 entries of genetic diseases and over 15,000 gene mutations that are known to cause these diseases. The others would be chromosomal abnormalities, for example. Not specifically related to disease, but useful for understanding many diseases is the Human Protein Atlas. And this, you type in typically a gene name into the search window, and it'll give you links to a whole bunch of information about where different genes are expressed in their protein products. So here's an example of glial fibrillary acidic protein, GFAP, and not surprisingly, the protein expression is highest in the central nervous system. It's unclear why the female tissues and male reproductive tissues get high scores also, and there are some weaknesses to the way this was done, but nevertheless, it does give an excellent overview of both the messenger RNA expression and the protein expression, with links to antibody labeling and that sort of thing. So, given all those numbers, it's obviously impossible for any of us non-AI types to know all of these diseases for either exam purposes or for your career in particular, because we're not going to see a lot of them. So I think it's important to appreciate the principles rather than have a comprehensive knowledge of all the diseases in your head. So you need to keep in mind that all of the tissue constituents, whether they're brought in from outside or produced locally, eventually they need to be degraded and recycled and then disposed of or expelled. And if that disposal mechanism fails and there's accumulation of those materials, those waste materials or stuff that would normally be recycled, then that can result in a disease state. And some of those accumulated materials can be stockpiled in a non-toxic way, which I'll talk about later, or they may accumulate and become toxic. And in some cases, the aggregates themselves aren't toxic, but they can be markers of blocked metabolic pathways in other systems, which may ultimately interfere with normal cell function or survival. Now you'll note that I'm talking in a very general term. This can pertain to any organ system, but we're obviously most interested in the central nervous system. So if there's things that are being turned over, what are they and in what cell types? So a lot of the cell turnover in the central nervous system is difficult to study directly. There are a number of interesting publications from Friesen who looked at basically the carbon-14 isotope that was deposited in parts of northern Europe from radioactivity, using that as a tracer of proliferating cells. Most of the information, though, is based on experimental administration of tritiated thymidine into experimental animals, and then the thymidine is incorporated into DNA, and so this gives you an idea of what cells are proliferating and what cells are degenerating. And not surprisingly, younger animals have higher turnover rates during growth. But just to summarize very briefly data from mainly adult mice and also some data from humans, neurons in general are post-mitotic. There's a whole lecture worth of debate about whether dentate granule neurons fit into that category and up to what age. Astrocytes turn over at about 0.4% per day. Oligodendrocytes much more slowly, about 1/10 of that rate. The information from both rodents and humans seems to be on par for that. So in general, you probably won't replace all of your oligodendrocytes in a lifetime. Endothelial cells turn over at about the same rate as astroglial cells. And of note, some of these cells may live from 1 to greater than 10 years. Microglia, a little bit more slowly than that. Within the cells, there is a more considerable turnover of mitochondria. So obviously, as cells are turning over, there's DNA synthesis, and what happens to DNA and its turnover. So in addition to mitotic activity, there's unscheduled DNA synthesis. So this basically refers to repair of damaged DNA. A lot of that damage will come from environmental factors such as cosmic radiation or environmental toxins. In mouse brain experiments, the half-life of nuclear DNA is approximately 20 days. And that's not including cell proliferation. Again, separate lecture topic, but various neurological diseases are associated with faulty DNA repair, as is many cancers. Most of the nucleosides are recycled. Some of them are eliminated as uric acid, which, if that accumulates, can cause gout, and gout in itself carries an increased risk of neurodegenerative diseases. Ribonucleases degrade messenger RNA following protein synthesis. When cells die, most of the DNA is recycled locally, and about less than 1% of brain cell DNA will get into plasma as cell-free DNA. So with respect to protein synthesis, the table on the right, the yellow highlights show the amino acids and their transport pumps that are present in the blood-brain barrier. Most of these in the solute carrier SLC family. So basically, most amino acids are going to be imported from blood circulation to create proteins as the messenger RNA is transcribed. After the peptide chain is created at the ribosomes, then it goes into the rough endoplasmic reticulum, where post-translational modifications are done using a variety of different enzymes to attach oligosaccharides. And then from the rough ER, these are then go to the Golgi apparatus for further post-translational modifications, and then depending on what type of protein and what type of modifications, which often act as signaling molecules for sorting, the proteins may go into secretory vesicles where they are secreted, or they may simply dock with the cell membranes where they become cell surface proteins, or in the case of lysosomes, they're stashed away in the lysosomal membrane for future use in digestion of cellular materials. Lots of experiments using radioactive labeling of amino acids, typically with different, mainly with carbon-14 isotope, administering these to different age animals. And basically the older data suggests that a small proportion of proteins have very rapid turnover, less than a day. And in the brain, the majority of proteins are moderately long-lived with half-lives of greater than 10 days. And overall, in about 2 to 3 weeks' time, most of the protein has turned over. The proteoglycan modifications will typically turn over at roughly the same rate, in some cases a bit faster. In adult rats, about 5% of the protein in the brain breaks down every day. Of note, though, the proteins of myelin, including myelin basic protein, are much more stable, with typically taking about 3 months for 100% turnover. The current technologies allow us to look at thousands of proteins and phosphorylation modifications much more rapidly. And so this publication from this year looked at 9 different brain regions in mice, and as well as different organ systems, and basically came to the same conclusion that most of the proteome, roughly 2/3 of it, has a half-life of less than 10 days. And the most long-lived proteins are typically in the energy provision, basically mitochondrial proteins, myelin sheath, and chromatin assembly. And there are a couple of examples. So, for example, GFAP and synaptophysin have a half-life of about 15 days, whereas myelin basic protein is somewhere between up to a half year. The same sorts of studies can be done with glycoproteomic analyses. Lipid, similarly, has to turn over as the proteins are turning over. So most of the lipid in cells consist of the phospholipid bilayer and all of the embedded proteins as shown on the right diagrams. Different lipid components recycle at different rates, and there are some old radioactive tracer studies, but the modern lipidomic assessments suggest that, for example, the fatty acids are slowest to turn over in brain compared to other organ systems, but they're fairly rapid with a half-life of about 3 days. Lots of experiments have addressed myelin lipid very specifically. So myelin is a specialized concentration of oligodendrocyte cell membranes. And basically, I'm sorry, this has got messed up a bit, but the different constituents of myelin have different turnover rates, with cholesterol being and cerebrosides being relatively stable, and phosphatidylcholine being less stable. What's underneath the table is a statement that compact myelin is more stable than non-compact myelin. Moving on to different cell constituent, glycogen. So we know that the brain is dependent on a constant supply of glucose for its energy production, and that glucose crosses the blood-brain barrier using the GLUT1 transporter. And once it's in the brain milieu, some of it may be used immediately by cells of the brain. Other glucose molecules may be taken up by astrocytes and then incorporated into glycogen. And the diagram on the top right shows the structure of glycogen with a chlor protein called glycogenin surrounded by thousands of glucose molecules in chains with different branch points, and different enzymes are involved in assembling the glucose molecules along these branching chains. And in general, the glucose will be incorporated quite rapidly within 6 hours, and it tends to turn over very quickly, in particular as the neuronal activity in the immediate vicinity is ramped up. And so in general, again, these are old carbon-14 tracer studies, about 50% will turn over within a day following incorporation. Of note, it's possible to do in vivo glycogen analyses using magnetic resonance spectroscopy, and again using a slightly different isotope. So this is carbon-13 labeled glucose. This can be infused into animals or humans, and it's been shown in both that complete turnover of glycogen takes about less than a week. So just to summarize then, basically everything that our cells are made of degrades and must be either repaired or disposed of, and all of the repair and disposal mechanisms need some sort of recycling. And so every cell has a number of different different organelles for doing this. You have the autosome, lysosome complex, peroxisomes, proteasomes, autophagy and mitophagy, and then that's within cells, and things that are expelled from cells, again, separate lecture topic, but basically clearance of waste products from the central nervous system via the extracellular fluid and the paravascular fluid pathways, now commonly referred to as the glymphatics, where eventually extracellular waste from the central nervous system mixes with cerebrospinal fluid and then eventually gets either into the venous system or the local lymphatics. So autophagy is an orderly removal of unneeded cell constituents. This is lysosome dependent, and mitophagy is similar, where mitochondria are recycled, and phagocytosis refers to engulfment of extracellular materials for recycling. Notably, most of the phagocytosis in the brain is done by microglia, which are immune system cells, and when more debris is present, so for example following physical trauma or an ischemic infarct, cells from the blood come in, circulating monocytes, and they become macrophages. It's important to note that astrocytes have been known for decades to play a limited role in phagocytosis, and this could be done by endocytosis or pinocytosis. And I'll talk a little bit about that Lysosomes are small membrane-bound organelles. They have over 60 different enzymes. They're designed to digest debris, both from inside and outside the cells. They're formed in the endoplasmic reticulum and Golgi, as I outlined earlier. And when needed, they fuse with the endosome. So that's the material that's been surrounded, so the cellular organelles that or pieces that are no longer needed within the cytoplasm, or with a phagosome, so something from the outside. Once that membrane-bound organelle is complete, it fuses with lysosomes. The enzymes within lysosomes are released and mixed and activated with the contents of the autophagosome or the external phagosome, and all of that's degraded and eventually recycled. Exocytosis. So some of the degraded material may be then recycled within the cytosol. Others is spit out, and we typically so that spitting out of material is called exocytosis. Basically, we've got a vesicle that contains waste, it fuses with cell membrane, and it releases it to the outside. Neurons and astroglia, exocytosis is extremely well studied in the synaptic vesicle and release and recycling context rather. But the lysosomal contents can also be released. Some lysosomes, it's been shown recently, are not necessarily ones that have been fused with autophagosomes. Some of them may be, the enzymes may be used locally outside of the cell for synaptic remodeling, and so this may be important for plasticity. And similarly, reactive astrocytes can release signaling molecules and other enzymes that are necessary for tissue remodeling following injury. So peroxisomes are small vesicles. They also contain a large number of enzymes. Their main functions are for hydrogen peroxide production, wherein the hydrogen peroxide is used to oxidize a variety of cell constituents, in particular very long-chain fatty acids. They also are capable of responding to oxidative stresses where there's too much hydrogen peroxide, for example, and so they can take up hydrogen peroxide and degrade it via the catalase enzyme, which is one of the main enzymes in peroxisomes. I mentioned that they're important for oxidative breakdown of various fatty acids. They also synthesize plasmalogens, which are glycerophospholipids, very important in myelin sheaths. The proteasome ubiquitin system, I think anybody who reads about neurodegenerative diseases has a fairly good understanding of this system. So these are basically protein complexes present within the nucleus and in the cytosol, and they are have proteolytic activity. So if you have a protein that's been tagged with ubiquitin as a signaling molecule, that protein enters the proteasome and is chopped up into peptides. There's two different types of proteasomes, one that is dependent on ATP energy and ubiquitin, and a smaller type of proteasome that is independent of ATP and ubiquitin. And so this is for degrading oxidatively damaged proteins. These are, as I say, very known to be important in neurodegenerative diseases. So lots of different cell constituents, all of them being broken down and recycled and regenerated at various paces, lots of recycling tools. What happens with all this stuff? Well, in normal state, and in particular in long-lived cells like neurons, all the cells have landfill sites, garbage dumps, and just like cities do, because you can't recycle and reuse everything. And so the two main types of landfill in cells are lipofuscin, which are highly oxidized macromolecules. So these lipofuscin aggregates shown on the middle bottom as this yellowish granular material. Here's an toluidine blue semi-thin section and an electron microscopic view. So these are a mix of protein and lipid with a small number of metal cations and sugar residues. They can't be degraded or thrown out, and so they basically are stashed in the back closet of the cell. The term spheroid appears in a lot of the literature and in some diseases. It's probably essentially the same stuff. It's just slightly different compositions in different cell points, so cell types rather. So spheroid and lipofuscin are biochemically interchangeable. One is referred to in a normal context, the other in a disease context. Corpora amylacea are these lamellated pearls of the brain, and these are typically within astrocytes. Most of the corpora amylacea are polyglucosan or glucose polymers, more than 90% of that, and there are small proteins within them. And the general hypothesis is that toxic proteins that can't be degraded or eliminated are wrapped up in sugar in order to make them less offensive. These stain well with the PAS method. In neurodegenerative diseases and around areas of damage, there's more breakdown and therefore more corpora amylacea. And their presence reflects, again, this capacity of astrocytes in their limited phagocytic capability. So they may be able to ingest material but not necessarily digest it. Some of these corpora amylacea are eventually expelled from astrocytes, and they can go through the CSF and then eventually be recycled in macrophages. And you'll note in almost every brain, they're easier to find around blood vessels and in the subpial region, possibly reflecting the fact that they can be released into the fluid points from there. So finally, on to diseases, and specifically storage diseases. So how do we categorize these? So you can consider the metabolic pathway involved, whether it's lipids or amino acids, energy supply, by the organelle involved, whether it's lysosomal, peroxisomal, mitochondrial, etc., by the end result, whether stuff accumulates in neurons and they are identifiable microscopically and then eventually die, whether there's early onset epilepsy, whether the brains shrink, whether the white matter is damaged in the form of leukodystrophies, or, and these aren't exclusive criteria, by the inheritance, whether it's X-linked, autosomal dominant, autosomal recessive. Many of these diseases have eponymous names reflecting the people who describe them. So in general, storage material refers to aggregates of protein, lipid, or carbohydrate, and this may reflect either indigestible material, which is phagocytosed and accumulated, and non-toxic. So I mentioned lipofuscin. Hemosiderin is another example, mainly in macrophages, but also in astrocytes. Enzyme deficiencies which are almost all genetic at their root, whether they're de novo mutations or inherited ones, that leave to the geneticists to sort it out. Or you can see storage in the context of environmental changes, diet, exposure to toxins. Now, whether we call something storage, that in some respects is arbitrary, in the sense that if we can see it at the light microscopic level and something is bigger or more obvious than it should be, then we can call it a storage disease. So storage disease isn't in scientific terms particularly useful, but it does encapsulate these concepts of waste products and failure to deal with them properly. So why do so many of these things present in youth? Well, this diagram from a recent publication focused on proteostatic stress. So this is the turnover of proteins and the stress that that creates. And not surprisingly, during the period of brain growth, whether it be synaptogenesis or myelin production, which are rapid in the first decade of life as the brain is doubling in weight, or actually almost tripling in weight, from birth 450 grams to somewhere around 1400 grams at 12 to 15 years. So during this period of rapid growth, there's more need for production and turnover of protein, and this can create stresses on the recycling systems. And so if they're not up to snuff by virtue of enzyme deficiency, for example, this can result in brain disease. In the old age, the neurodegenerative diseases, you have similar kinds of environmental stress that can lead to protein misfolding and various diseases. So this diagram gives you the idea that the storage and metabolic diseases of childhood are at least thematically related to many of the neurodegenerative diseases in the elderly. I mentioned how there's no way of knowing all of this stuff. One of my favorite books on the subject of metabolic diseases is this 1995 text by Chervos Navarro. It's obviously not in print anymore, but I was looking at it for purposes of doing this lecture, and I think although the genetics and some of the biochemistry is a little bit outdated, I think the discussions of morphology of these diseases is still relevant. So on to a few examples. Glycogen storage being one of the simpler ones. This is called Lafora disease, after the pathologist who described the accumulation of the polyglucosan bodies. Most of these are caused by loss of function mutations in these two genes, which encode proteins that are called laforin and malin. And basically, these two proteins form a complex that is important for assembly of glycogen. And if there's mutation in either one, you'll get abnormal branching of the glucose molecules in the glycogen assembly, and usually hyperphosphorylation as well. And the problem with that is that the glycogen, once it's been assembled in an abnormal way, can't be disassembled easily because the enzymes for disassembly are designed to work on the normal structure of glycogen. And so the end result is that glycogen accumulates in these polyglucosan or Lafora bodies in the brain, liver, myocytes, myoepithelial cells of the sweat glands. The typical clinical presentation is seizures followed by neurological decline and death in approximately a decade after the onset of the disease. Moving on to peroxisomal dysfunction, there are a number of diseases that are related to this. Some of them are related to the failure of peroxisomes to be produced properly, in which case you've got multiple protein and enzymatic deficiencies and abnormal or absent peroxisomes. This would include the Zellweger syndrome, the neonatal adrenal leukodystrophy, and infantile Refsum disease. In other cases, you'll have a single gene defect that results in one of the proteins or enzymes in peroxisomes being dysfunctional. And in this circumstance, electron microscopy would show normal peroxisomal morphology. One of the most severe forms of peroxisomal disease is the Zellweger syndrome. This is typically either a homozygous or a compound heterozygote mutation in the PEX1 gene. These affected children have distinctive facial features at birth and bone abnormalities. The brain may be small, it may appear to have pachygyria, and electron microscopy will show abnormal lipid membranes and autophagosomes because the peroxisomes are not properly degrading the very long chain fatty acids. They end up in lysosomes and can't be digested properly there. So the PEX1 is important in the importing proteins into the peroxisomes. And of note, so going to the human protein atlas, you can see that the PEX1 expression, there is some mRNA, but the protein expression is actually rather low in the brain, and it's present in most other organ systems. And so the point here is that simply looking at where protein is abundant doesn't always directly predict what organ systems might be affected in a disease state. The mucopolysaccharidoses are a large family of diseases. These are associated with mutations in various genes that result in deficient enzyme activities that are important for breaking down the glycosaminoglycans and heparan sulfate. And the most common ones, and keep in mind all of these diseases are rare, so the most common ones are Hurler, Hunter, and Sanfilippo syndrome, and I'll just show some examples. So this is a MPS 1 case, a female who had presented with a large head in infancy, severe hydrocephalus. She was shunted, diagnosed with Hurler syndrome due to an IDUA mutation. She received a stem cell transplant at 1 year age, which normalized her circulating cell enzyme activity. She continued to have seizures. Her intellectual impairment was not reversed, but she lived quite a long time, died at 20 years of age. The photograph shows her coarse facial features. Her brain was abnormally small. The ventricles were small because her shunt was still functioning. And keeping in mind that she did have a stem cell transplant which was functional, she had only a limited number of neurons that had abnormal storage material that stained with Luxol fast blue, and also abnormal PAS staining storage material. The IDUA is abundant in brain, mainly in neurons, and this image on the right shows one of the examples of the tissue microarray information available on the Human Protein Atlas. Here's an MPS II case, a 12-year-old male with severe neurologic decline. His ventricles were large. You can see the enlarged perivascular spaces in the atrophic white matter. Histologically, his dura was very thick, and this will in turn reflect on the abnormal cerebrospinal fluid flow that can cause hydrocephalus. Perivascular spaces are enlarged and filled with macrophages that don't stain with the Luxol fast blue method. You can see relatively normal myelin in the white matter. And here's the neocortex on the left and the cerebellar cortex on the right, with LFB stained storage material within neuron cell bodies and within dendrites of the Purkinje neurons in the molecular layer. MPS III, Sanfilippo disease, is actually several different mutations and enzyme deficiencies which have onsets at different ages. Histologically, these are very similar to the previous two types, with LFB accumulating material in neurons, and these show the dendritic expansions. So in the cerebellum, more commonly than in the cell body, you see these dendritic expansions. MPS IV is brain typically isn't directly infected, but as a neuropathologist, we can sometimes see these cases because it's a skeletal disease. And had this autopsy a few years ago. 33-year-old male who had severe scoliosis, short stature, cognitively normal. He had been diagnosed with atlantoaxial instability at 16 years of age. That was associated with a fusion, but eventually that fusion broke down. He had a respiratory arrest while at the dentist, and this is his medulla on the right, his mid-cervical cord on the left, and you can see the atrophy of the upper cervical cord, which was compromised during the dental procedure. Gaucher disease is a mutation in the GBA1 gene, which is glucose cerebrosidase in lysosomes. Different types depending on whether the nervous system is involved. This is a perinatal case. We saw it born at 35 weeks following abnormal ultrasound, died in a couple of hours. Macrophages in the perivascular spaces, slightly abnormal appearing brain, macrophages throughout the cortex, PAS stained material, mainly in the cortex and perivascular spaces. The ultrastructure was not the typical Gaucher cells. Here's a type 3 case, a 22-year-old female who had seizures for 4 years. You can see these abundant macrophages with this lamellar inclusion material around the perivascular spaces. GBA1 is basically in all cell types in all organ systems. Krabbe disease, otherwise known as globoid cell leukodystrophy, is a lysosomal deficiency of galactocerebroside beta-galactosidase due to the GALC mutation, and basically membrane lipids are not broken down, and you get accumulation of an abnormal lipid called cyclosine, which is neurotoxic. Here's an example of a 19-week fetus There was a known family history, abnormal ultrasound prompted termination, and you can see abnormal macrophages within the spinal cord. These are examples from a young girl who had developmental regression at about 2 years age. The sural nerve biopsy showed these abnormal lamellar inclusions in the Schwann cells. When she died at 9 years age, the Gaucher cells could not be identified. She had this spongiform destruction of cortex and these kind of non-specific PAS staining macrophages in perivascular spaces. Here's another a 12-year-old boy with severe developmental delay. You can see the white matter degeneration, macrophages, again, no globoid cells. And what these show is that these macrophages with the so-called characteristic inclusions don't necessarily persist in long-term survivors. Finally, the neuronal ceroid lipofuscinoses, different diseases depending on the gene, categorized by the gene mutation and the age of onset. These genes the proteins are expressed in all organ systems, and another feature of the Human Protein Atlas, you can see which organelles these would normally be in this case, Golgi and lysosomes. Here's an example of an atrophic brain with negligible myelin. This is in a young boy, like 10 years old, and abundant PAS staining macrophages, and on and on. So in the past, typically what would happen is you'd have a storage disease suspected, then they'd get an enzyme analysis, and there'd be a biopsy of some tissue with electron microscopy, and then eventually an autopsy to prove the disease. Currently, though, typically most of these cases are identified by genetic testing, and they never come to autopsy or even biopsy, and so that's part of the reason we don't see these things very often. In some cases, you'll see modification of diseases by virtue of treatments, either enzyme replacement or marrow transplant. The circumstances where autopsies might occur are when there's tissue donation for research of the disease, unexpected medical legal deaths, or these fetal or perinatal cases that aren't expected. There's lots of phenotypic variability, and some of this is going to be related to the degree of enzyme activity, the specific mutation, modifier genes, epigenetic modifications, environmental influences, including diet, etc. And in general, although we talk about these as storage diseases, what you have to remember is that the classic storage materials might not persist in chronic cases. If those inclusions are present in macrophages, a lot of those will eventually emigrate and or the neurons will die off, and so you won't necessarily see an abundance of storage material. The so-called specificity of electron microscopy that's an overstatement by far. There's lots of different morphologies that are possible, in particular with the ceroid lipofuscinoses. And again, keep in mind that the phenotype is not obviously explained by the relevant gene and protein distribution. Again, this is from the same article that I showed the graph of young and old. There is some overlap in the concepts of these pediatric storage diseases and the eventual neurodegenerative diseases, with failures of the recycling system, accumulation of something, and eventual toxicity. And those are a few additional references, and I shall stop there.
Thank you, Mark. So at this time, we're going to move into the Q&A. You can submit your questions in the chat box, or you can unmute to ask a question. When submitting a question in the chat box, please make sure it's sent to everyone so that we can all see it. And to unmute, select the microphone in the lower left, and please remute after you have asked your question.
Yeah. So I can see the chat box. So the first question is, in the mucopolysaccharidoses, isn't the intraneuronal storage material, ganglioside? So the short answer is yes. And that's one of the reasons why it will stain with the Luxol fast blue method, which basically stains the gangliosides in normal myelin as well.
I don't see any questions, so I'd like to ask my own question. I just want to know, like I also saw these diseases a lot more in my early training, and then they obviously disappeared as things went on. But now, what should like a year or two medical student know of what you just discussed today?
Well, I think and that's part of the reason why the first half was devoted to more to the concepts around the fact that nothing in the cell is stable. Everything is turned over and disposed of or recycled by different organelle systems. And in many cases, if those recycling or disposal mechanisms don't work, you can end up with tissue toxicity. And in some cases, or some circumstances, those failures may lead to microscopic aggregation of materials. And so when we see a lot of whatever LFB staining stuff or PAS staining stuff, we call it a storage disease. If we at the old age end, if we see abnormal ubiquitination, we call it a neurodegenerative disease. But these things all sort of fit in the same conceptual spectrum. Different types of cell constituents are broken down and disposed of in different ways. And some of them will aggregate into big blobs and others won't necessarily. But they're using the different tools we have at our disposal, we can identify them.
Okay, thank you. I'll let anyone else ask a question.
All right. Are there any additional questions, Dr. Oviedo, or are we good to wrap up the session?
I think I don't have any more questions. So.
Okay. Well, thank you again for joining today's AANP Teaching Rounds presentation. We would ask that you take a few minutes to complete a short evaluation. Completion helps ensure accurate reporting to the accreditation board. A link to the evaluation will appear on your Path LMS screen upon the conclusion of the Zoom meeting, or you can navigate back to the main course and select the evaluation option. Once you've completed the evaluation and selected your credit amount, please then select the appropriate certificate based on your credentials. The PowerPoint slides and recording will be posted to the AANP website in the next week. Thank you again to Dr. Del Bigio for an excellent presentation. This concludes the session for today.
Bye, everybody.
Have a good day. Bye.